So today I'm going to be talking about flexible low-density SNP panels and in animal breeding. So I'm going to start off by telling you a little bit about me, because it really tells you why I'm talking about this subject area and some limitations to what I can speak about today. So until 7 months ago, I was the global genomics manager for Aviogen, one of the world's largest broiler breeders. And I was responsible for taking the genomics program from an R&D phase through to full implementation. Now, during this process, we moved from processing about 2,000 samples a year all the way up to more than 15,000 samples a week. Now, this was a mixture of high-density and low-density SNP genotyping, but we had a LGC SNP line, and we're generating about 1 million data points per week. So that's why I'm here to talk, but unfortunately I can't go into a huge amount of specifics for what we were doing, but I'll talk about it in very general terms. I'm now the director of the National Avian Research Facility, or the NARF, based at the Roslin Institute, and we've got a CapEx bid in for an LGC SNP line, and we hope to implement some of the technology in the NARF, and we'll be initiating a research program looking at the genomics of commercially important traits in avian species. So what I'm going to talk about today is the low-density SNP panels. And I'd like to sort of suggest that they now can be used as a very basic tool in animal breeding. And we used them a huge amount when I was at Aviogen, and I'd like to share some of those experiences in a very general way. So we can use them in genomic selection. We can also use them in population management through Pedigree Reconstruction. And our plans at the NARF are to bring these two elements together and add value to some of our avian model animal populations. So first of all, genomic selection. Now, I'm sure that I don't have to talk about the promise of genomics to this audience. And really now, it's not really a promise, because we're really seeing the realization of the long road along the R&D to see actual increases in accuracy of genomic predictions and increase in progress through those increase in accuracy and reduced generation interval. We can also use genomics to improve the selection for traits in commercial environments, so looking at pedigree and commercial type of environments. And we can use it for management of genetic pools. So this is what the promise was. When we started the genomics program in Aviogen, then we were actually, we had a few challenges. So in 2004, we had the first draft genome for chickens, and it was actually the first commercial species to have a full draft genome. The challenge that we faced was that there wasn't a commercially available chip. The pig breeders and cattle breeders very rapidly produced commercially available chips after the publication of their genomes. There were a number of proprietary chips that were developed, but these were fairly expensive, and because of the cost associated with developing them, there was always questions about whether they were actually providing all the information that was required to capture genomic information and predict genomic breeding values. So at this meeting in 2009, we started along the road to develop a commercially available high-density chip panel. And in 2012, that was released. It was approximately 600,000 SNPs, and it was developed in collaboration with the EW group, so Aviogen, part of the EW group, the Roslin Institute and Affymetrix, and funded by the UK's BBSRC funding body. Now, I lost a few bets along the way when we developed this SNP chip, because my desire was that we were going to use all this information, but once we had all the information, we were able to evaluate that we didn't need 600,000 SNPs. So today, a lot of breeding programs are developing lower density SNP panels, and the EW group now uses approximately 50,000 SNPs across a number of different breeding programs. So now that we had the really high density SNP panels, we're able to look to see how many we really needed. And the research going on at the time was suggesting that we were reaching inflection points in the tens of thousands rather than in the hundreds of thousands. So this data from Holstein suggests that we're capturing all the predictive accuracy by less than 10,000 SNPs. Now, obviously, the genetic architecture of a Holstein is very different to a chicken, and you know, there's a lot of variation in terms of the sweet point for the number of SNPs when we consider the genetic architecture. If we go to human populations, this was something that I really held on to, that when we actually increased the training sets, we were able to utilize more of the information from a higher density SNP panel. Although the inflection point doesn't really change dramatically, we can actually capitalize on that additional information to some extent. What we were seeing when we were trying to implement a genomic breeding program was that, you know, this is a graph that everyone's familiar with, the sequencing costs and the deviation from Moore's law, the dramatic reduction in cost per base pair for sequencing. Now, I think that everyone would agree that in terms of the SNP panels that we had access to, this was probably also the case, that our cost per SNP was going down and down. However, the way that the SNP panels were being developed, it wasn't really reducing our cost per individual. It was actually providing us with more and more information on those individuals. So for the implementing a genomic breeding program, that was a bit of a challenge. We did some sums when we had just the high density panel, and we estimated that to implement genomics, to genetically improve a single bird breed, which is a four-way cross, would cost in the region of £15 million per annum, and that was an ongoing cost. That was considered to be prohibitively expensive, so that we were tasked with trying to rationalise those genotyping costs. And this is where generation of low-density SNP panels comes in. So we were able to capture some funding from the EU. That may finish for us in the UK very shortly. And we collaborated with Jack Deckers at Iowa State and John Hickey at the Roslin Institute to deliver a demonstration project which tested the utility of evenly spaced low-density SNP panels. I'm not going to talk about the technology associated with doing the actual imputation, but I want to touch on some of the results that we were seeing and the importance of those results for our breeding program. So we had a data set where we genotyped at two different low densities, one being 377 markers and 766 markers. We also had high density genotypes on these individuals, and we were able to compare the true genotype with the imputed genotype. So the correlation between imputed and observed genotypes per SNP was approximately 94%. If we look across SNPs within individuals, our correlation between imputed and observed genotypes was about 97% for a 766 density, low density SNP panel. So we felt that we were doing pretty well when it came to imputing those genotypes. And we were further encouraged when we actually went on to look at the impact of these imputed genotypes on the accuracy of genomic breeding values. And we could see that the accuracy on the imputation was really the upper limit on any loss of accuracy on the genomic breeding values. So for a 766 SNP panel, we were losing less than 2.5% on at the very maximum in accuracy. And if we compare the cost using LGC platform of genotyping for 766 versus some of our high-density SNP panels, then you can see that the impact on a large breeding program can be fairly significant. What this doesn't consider is the very changing environment that we work in. And once the breeding program was able to identify that genomics was going to be effective in delivering increase in accuracy, then there was a fairly large-scale implementation across the sister companies of EW Group, which enabled the EW Group to negotiate on volume. This meant that imputation wasn't such a driving force anymore. However, moving back to the academic sector, I can see that we're always trying to drive down the costs of our research grants, and the imputation becomes extremely important for some of our projects. So moving on to using low-density SNP panels in population management, I want to talk about academic and commercial pressures on bird management. Now, for the commercial situation, there's a kind of paradox in terms of our estimation of fertility, male fertility. So the standard way that a breeding program would maintain the pedigree would be to have a pen with one male and a harem of females, maybe 10 or 12 females. He's got a fairly cozy lifestyle. So we measure his fertility across these 10 to 12 females. However, when we want him to work commercially, it's a very different situation. The ratio still remains the same, about 1 to 10. However, he has male competition to contend with and female choice. So being able to phenotype him in a more commercially applicable scenario can be extremely important. For us in the UK, thinking about how we house experimental birds, we are governed by the ASPA, which is the Animal Scientific Procedures Act of 1986. And this is almost, it's a real challenge to manage a pedigree breeding program. Essentially, to maintain pedigree, you have to house a single male with a single female, unless you're going to use genomics to disentangle the pedigree. And that's not great for bird welfare, because males and females don't really interact like that in bird environment. And also it's a challenge to try and cost any breeding program for us in experimental facilities. So that's where pedigree reconstruction becomes a really useful tool for us. What we need is genotype information on the parent-offspring trio. We use evenly spaced low-density SNP panels, a high minor allele frequency, low LD between markers, and we can estimate, we can reconstruct pedigree with as little as 70 markers. The algorithm is based on minimizing the Mendelian inconsistencies between parent offsprings and the parent offspring trio. So essentially the way it works is that we first set the male to minimize those Mendelian inconsistencies. We use the male because there's fewer males, and so our chance of getting it right first time is much improved. Based on that parent male and child relationship, we identify a dam which minimizes the Mendelian inconsistencies. We then circle around that process again to home in on the true parent-offspring trio. And we've been able to estimate error rates of less than 0.5% using this process. Now, commercially, it can be a bit of a challenge because when you're making decisions in a breeding program, it's fairly early in the life of the bird, so you need those rapid turnarounds. And that's where the LGC SNP line is extremely useful. And I should say, I was really interested to see the other talks home in on DNA extraction. For birds, we're very privileged to have nucleated red blood cells. So we don't even extract the DNA, we just burst the cells and precipitate all the nasty stuff. So we don't have the challenges of DNA extraction, although if we did, I think processing 15,000 samples a week would become a real headache. So in terms of combining these two different approaches, which is what we're hoping to do in the NAF, I think that we'll be able to generate some useful projects. So a little bit about the NAF, the National Avian Research Facility at the Roslin. We consider it a gateway to poultry research. We have the infrastructure, so we have two brand new facilities, the Greenwood building, which is our conventional bird facility, and the Bumstead building, which is our SPF facility. We have the people, so the critical mass of people working on avians, working on areas from genetics through to immunology and even down to genome editing and production of genetically modified birds. And we have the bird resources. So some of the bird resources that we have access to, we currently house the Institute of Animal Health Compton inbred lines, which are characterized for differential resistance and susceptibility to a range of bird diseases. This is largely mediated through the MHC, but there are other genomic regions involved. We have a number of transgenic reporter lines, which can be used in functional genomic studies and developmental studies. We also have access to a couple of model lines, and we're hoping to develop further model lines to increase our portfolio. These are closed, outbred SPF populations. And this is really where we're hoping to improve the utility of some of our bird resources. So in these outbred populations, we have funding proposals being prepared that the objective is to produce a sequence-based SNP library. We will sequence all the males within the population, so that's all the sires, the grandparents and great-grandparents. The females will be genotyped on a low-density SNP panel using the SNP line that we have CapEx in for the moment. And this will be a phased SNP panel. Using that information on the parents, any progeny from that population will have the ability to fully impute their sequence genotype. So from a low-density SNP panel, we'll be able to impute up to sequence. Our experience tells us that we can do that with a very high accuracy of about 97% plus, and it will be fairly cost-effective. So essentially, by genotyping on a low-density panel, people buying from our facilities will be able to impute up to full sequence. So we hope that this will really add value to the birds lines that we have at the moment, but also the resources that we're hoping to develop in the future. And thank you for listening to me.